Okay, okay, great. I think we still have like about 7 minutes to start, I believe.
Yes, sir.
Yes, yes, sir.
Welcome, sir.
Yeah, thank you very much.
Ma'am, should we wait for 5 minutes then?
Yes, we should wait. We should
Oh,
wait.
okay, ma'am. Okay, ma'am.
Validi, can we start?
Uh, 12, we should start at 2:30.
Okay, okay.
ও কিচ্ছু করবে না, ও কিছু করবে না, ও কিচ্ছু করবে না।
Ma'am, your microphone is on.
Anyone please check whether the YouTube streaming has started or not? Shomam?
Yes, ma'am, let me check once. Yes, ma'am. Streaming is started, ma'am.
Okay, thank you. So shall we start?
Yes.
2 more minutes. Okay, I'm starting, okay.
Yes, ma'am.
Good afternoon, everyone. As we all know, since 2020, COVID 19 has changed the whole scenario of the nutrition focused programs in our country. Due to this, essential services for the providing to the people who are dealing with nutritional problems like your ICDS programs, Angadwadi services, all have been disrupted. Due to this, the Ministry of Women and Child Health have launched recently again in 2021 February Mission Poshan 2, that is nourishing the future of India. And these various programs have been included, which we have been following for these 7 days consecutively, and the whole month also will be celebrating it. And our university, the Department of Home Science, has been celebrating this Poshan Ma since its inception, that is 2018. Last year also we had organized the international webinar series where various people from fields of food and nutrition, food science, food chemistry, and other allied subjects have been invited and discussed on various fields of these nutrition problems and strategies to overcome the various health issues related to nutrition. So today, for the second international webinar series, today is the last day of our webinar. So we have today also with us two eminent persons from the fields of nutrition and related allied subjects who will be enlightening us with their knowledge in their own fields. So I welcome the first speaker, our Dr. Bhimal Karani. He is professor of nutrigenetics and nutrigenomics from the University of Reading. Now I welcome Ms. Trina to introduce her.
Yes, ma'am. Thank you. Now we move on to the first lecture session. Our first speaker is Professor Bhimal Karani. Professor Karani has an interdisciplinary academic background with qualifications from molecular genetics, bioinformatics, statistical genetics, and genetic epidemiology. He joined the University of Reading after his postdoctoral training at the MRC Epidemiology Unit, Cambridge, UK, and University College London, London, UK. He has received uh, advanced training in epidemiology at the London School of Hygiene and Tropical Medicine. He has published extensively in areas related to nutrigenetics and non-communicable diseases, and presently has more than 80 peer-reviewed publications and book chapters. He won the UK Nutrition Society's Silver Medal Award for the year 2020 for his contribution to the world of global nutrition and for his research on gene-nutrient interaction collaboration towards implementing multi-ethnic population-based nutrigenetic studies of obesity in lower middle-income countries. His primary research interests focus on the investigation of gene-nutrient interaction on metabolic and cardiovascular disease-related outcomes using combined approaches of genetic epidemiology, statistical genetics, and molecular biology. Professor Karani is also a study coordinator of a large international collaboration, DCARDIA collaboration, principal investigator and founder of the Gene Nutrient Interaction Collaboration Academia Ambassador for Food Matters and Careers UK, and external reviewer of research output for Research Excellence Framework. He will be speaking on the topic entitled Precision Nutrition Strategy for Prevention of Cardiometabolic Disease. I would request all the participants to turn off their audio and video during the lecture session, and if there is any questions, you can write down your questions in the chat box. Welcome, sir. The platform is yours now.
Thank you so much for a wonderful introduction. So let me share the screen. Are you able to see my screen?
Yes, sir, we can see.
Okay, good. So, a very good afternoon to everyone. Thank you all so much for joining the webinar today. And thanks to the organizing committee for inviting me to give this lecture on precision nutrition strategy for the prevention of cardiometabolic diseases. So what I'm going to cover in my talk today is basically about like how the nutrigenetics findings could be translated to precision nutrition. So when you talk about the health of an individual, the health depends on various factors. It is not just the diet. There are various other factors such as the genome, the transcriptome, the proteome, metabolome, epigenome, and microbiome. So they all interact with the diet that you're consuming. And this interaction is the one that is contributing to the development of cardiometabolic diseases such as obesity or diabetes or cardiovascular disease. But in order to prevent obesity or diabetes or cardiovascular disease, the most important thing is to develop a diet which could be individualized or personalized for an individual. And that should be based on all these factors together. So the key question is, how are we going to integrate the data from all these multi-omics platforms and the dietary intake and the lifestyle factors? So that integration will require a machine learning approach, or otherwise what we call as the artificial intelligence platform. Because this platform is the one that's going to determine the implementation of the personalized diet. So this is exactly what I'm going to cover in my talk today. So to begin with, the gene-environment interactions on cardiometabolic diseases. So there are various factors which can predispose an individual to become obese or diabetic. And one of these factors is basically the lifestyle factors, which includes the dietary intake and the physical activity. And in addition, we also have the genetic factors, which can genetically predispose an individual to become diabetic or obese. So it's basically the interaction between the genetic and the lifestyle factors which contributes to the development of these cardiometabolic diseases. Or in other words, genes load the gun, but the environment pulls the trigger. So this metaphor clearly explains the intricate interplay between the genes and the environmental factor. And this is what you call as a gene-environment interaction. So with this background, let's have a look at what is a genetic variation. So for those who do not have a background in genetics, so I'm just going to show you a couple of slides giving you an overview about this genetic aspects. So we all carry different kinds of genetic variations in our human genome. Say, for example, this person has got this DNA sequence. And say, for example, this population has got like 5 individuals, for example. And you can see that 99.9% of the DNA is identical in all the individuals, where 0.1% carries all the genetic variations. And if you look at these sequences, you can see that some people carry T and some people carry A. So there's a change from T to A. So this change from one nucleotide to another nucleotide is what you call as a single nucleotide polymorphism. And depending on whether you carry T or A, so A is basically the genetic variation. And people who carry T do not carry any copies of the genetic variation. So depending on whether you carry T or A, you can classify individuals into 3 different categories as 0, 1, and 2. So those who do not carry any copies of the genetic variation, they are classified as 0 copy individuals. And those who carry 1 copy of A, that is 1 copy of the genetic variation, and 1 copy without the genetic variation, and those individuals are classified as 1 copy individuals. And those who carry 2 copies of the A or the 2 copies of the genetic variation, they are classified as 2 copy individuals. So for a given genetic variation or for a given SNP, there can be only 3 groups of people. Some people carrying 0 copies, some people carrying 1 copy, and some people carrying 2 copies. And generally, people who carry 2 copies, they have increased genetic risk. So here I'm talking about one SNP, one single nucleotide polymorphism. So please remember that our human DNA has got nearly about 4 to 5 million genetic variations, or 4 to 5 million single nucleotide polymorphisms. So which means that if you carry 2 copies in all these genetic variations, which means that you are basically accumulating your genetic risk. Your genetic risk is increasing depending upon the number of 2 copies that you carry in your human DNA. So what is this genetic risk? What do you mean by genetic risk, and how is that related to the development of a disease? So looking at that, if you carry a 0 copy, it means that you have low genetic risk because you don't carry any copies of the genetic variation. So which means that you have decreased risk of developing the disease. But if you carry 1 copy, it means that you have a moderate genetic risk, which means that you have moderate risk of developing a particular disease such as obesity or diabetes. And if you carry 2 copies, it means that you have a very high genetic risk, and which means that you have increased risk of developing the disease. But please remember that just because you carry 2 copies, it doesn't mean that you are destined to become obese or diabetic. You can still overcome that genetic risk by modifying your lifestyle. So how can you modify your lifestyle? That is, consuming a healthy diet and doing more levels of physical activity can overcome the genetic risk of obesity or diabetes or cardiovascular disease. So this is the underlying basis on which we do all the nutrigenetics and nutrigenomics research studies. So with this background, let's move on to what is nutrigenetics and what is nutrigenomics. So nutrigenetics refers to the study of the impact of the genetic variations on chronic disease outcomes in response to a particular diet. Say, for example, if I have a high genetic risk, that is, I'm carrying 2 copies, and I'm consuming a completely unhealthy diet, that is, high-fat diet. So the question is, what is the risk of developing obesity? So in order to answer that question, I need to look at the interaction between my genetic risk and my dietary intake. So this study of the gene-diet interaction is what you call as a nutrigenetics study. Whereas nutrigenomics is completely a different area of research. As the name implicates, omics means expression, genomics refers to gene expression, and nutrigenomics refers to the impact of the nutrient on gene expression. That is, what do you mean by gene expression? That is how the DNA is converted to RNA and how the RNA is converted to protein. So for the conversion of DNA to RNA and RNA to protein, you need to consume certain foods. And this study of how certain foods or how your diet is influencing your gene expression is what you call as nutrigenomics. And findings from nutrigenetics and nutrigenomics are essential for the implementation of personalized nutrition. So personalized nutrition is where you're developing an optimum diet for an individual depending on that individual's genetic makeup. For example, if I carry 2 copies, what is the kind of diet I should be consuming? Or if I carry 0 copy, what is the kind of diet I should be consuming? So that diet is what you call as a personalized diet. So first, looking at the nutrigenetics, as mentioned before, nutrigenetics is nothing but exploring the gene-diet interactions. So as part of running all these nutrigenetic studies, I'm leading 2 large collaborative network. One is basically the DCARDIA collaboration, which was initially funded by the British Heart Foundation. To date, we have nearly about 35 studies comprising 100,000 individuals from different parts of the UK, US, and European countries. And with access to the UK Biobank, which has got 500,000 people, and with access to various consortia-based studies, we have a sample size of nearly about 1.9 million people. And these are some of the publications from the DCARDIA collaboration. So the DCARDIA collaboration, as you can see, it's focusing on the developed parts of the world. That is basically looking at the Western populations. So to look at the lower middle income countries or looking at developing countries, I've initiated another large-scale collaborative network, which is called the Genuine Collaboration. So Genuine stands for the Gene-Nutrient Interactions, which was initially funded by the British Nutrition Foundation. And these are the countries in which I've implemented the nutrigenetic studies for the first time. And what you see in the brackets are basically the funding organizations that have supported my research activities in all these countries. So let me start with some interesting findings from the DCARDIA collaboration. So the main objective of the DCARDIA collaboration is basically to explore the causal relationship between the exposure and the outcome. That is, we wanted to know whether getting exposed to a particular dietary factor or to a particular disease, does that have an impact on the development of other chronic diseases? So to explore this causal relationship, we used a genetic approach. So that approach is called as a Mendelian randomization. So let me give you an example. So the first study that we did was we wanted to explore whether obesity leads to vitamin D deficiency or whether vitamin D deficiency leads to obesity. Well, there are several studies which have shown that higher BMI is associated with lower 25-hydroxyvitamin D concentrations. And there are also studies which have shown that vitamin D deficiency is associated with higher BMI. So the question is, which is a causal relationship? Is it obesity leading to vitamin D deficiency, or whether vitamin D deficiency is causally associated with obesity? So we used the DCARDIA collaboration samples. We had a sample size of about nearly 42,000 individuals, and we were able to do this genetic approach, and we found that for every 10% increase in genetically instrumented BMI, that is, as the BMI increased by 10%, there was a 4.2% decrease in 25-hydroxyvitamin D concentrations, which means that higher BMI is causally associated with lower 25-hydroxyvitamin D concentrations. And the next study that we did was looking at the association, the causal relationship between vitamin D deficiency and cardiovascular disease. And we were able to show that higher genetic risk of vitamin D deficiency was causally associated with increased risk of cardiovascular disease outcomes, which means that improving your vitamin D status, or in other words, taking vitamin D supplementation, if you're vitamin D deficient, then that could be an effective strategy for preventing the cardiovascular disease related outcomes such as high blood pressure or hypertension. And quite recently, we also did a study in relation to cancer. We looked at 7 different types of cancer, and we found a causal relationship between higher vitamin D concentrations showed a protective effect against the development of prostate cancer. And in collaboration with the team in Indonesia, we were able to look at the association between the vitamin D deficiency during pregnancy in relation to the abnormalities in fetal growth, and where we were able to show that vitamin D deficiency during pregnancy was significantly associated with abnormalities in fetal growth. And recently, we also did a phenome-wide association study, which is nothing but where we looked at the association of obesity with 925 disease outcomes using the UK Biobank, that is using 500,000 people data. And we were able to show that the genetically instrumented obesity was associated with 33 disease outcomes, which were related to the endocrine problem or relating to the circulatory system or digestive system or even skin infections, including type 2 diabetes. And there has always been a question, is it depression that causes obesity, or is it obesity that causes depression? So to put an end to that story, we looked at the causal relationship between depression and obesity, and we were able to show that individuals who have high genetic risk for depression, they showed a significant causal relationship with obesity. So which means that depression causes obesity and not the other way around. And quite recently, we published data in nearly about 2 million people where we were able to show that higher milk consumption, that is consuming more than 1 glass of milk per day, was significantly associated with increased BMI. And now we are looking at the association between lipids, inflammation with vitamin D deficiency as well. So the reason why I'm showing this particular slide is just to highlight the fact that the main root cause for the development of various chronic diseases is basically obesity. That is, if you're able to prevent obesity, then you're indirectly preventing the development of various chronic disease outcomes. So preventing obesity through a personalized diet is very much essential. We have all been following the universal dietary recommendations for several decades, but we still see a significant increase in the prevalence of obesity or diabetes or cardiovascular disease, which means that the universal dietary recommendations are not working well for everyone. And that is exactly the reason why we are moving towards a personalized approach. That is, we are moving from one size fits all approach towards a personalized approach. So now let's have a look at the genuine collaboration, which focuses on lower middle income countries. The genuine stand for the gene nutrient interactions. It's been 7 years since I started this collaborative network. And the main objective of the genuine collaboration is basically to implement nutrigenetic studies for the first time in contrasting ethnic groups. The first nutrigenetic study was implemented in the Indian population, followed by a study in the Sri Lanka, and then in Indonesia, in Brazil, in Ghanaian population, Turkish population, and Peruvian population. And these are some of the other countries in which we are implementing the nutrigenetic studies, and in addition to the ongoing collaborations in various parts of the world. And these are some of the publications from the genuine collaboration in the last 5 years. So this is just to highlight the fact that the nutrigenetic studies were not available in developing countries before the implementation of the genuine collaboration. So the genuine collaboration is the first approach or collaborative network to implement such nutrigenetic studies in lower middle income countries for the first time. So let me share some of the interesting findings, how the gene diet interactions are happening in different lower middle income countries. So we all carry different level of genetic risk for obesity. Some people might be low genetic risk, and some people might be moderate genetic risk, and some people might be carrying a high genetic risk. But just because you carry a high genetic risk, it doesn't mean that you're destined to become obese. There is always a trigger factor, and that triggering factor is nothing but your dietary intake. Your diet is the one that determines whether you're going to become obese or not. Even if you have a high genetic risk, by consuming a healthy diet, you can overcome that genetic risk of obesity. So several studies were carried out in different parts of the world. So let me focus first on the South Asian countries in India and Sri Lanka. So in these two countries, like, we carried out these nutrigenetic studies, and we identified that the diets high in carbohydrates, that was a triggering factor for increasing the genetic risk of obesity. So those individuals who were consuming more than 500 grams of carbohydrates per day, that increased the genetic risk of obesity in Indian as well as the Sri Lankan population, because their kind of diet is almost the same. And the Indian study was done in South Indian population. That was a study in Chennai. And if you look at the diet in South India, which is almost the same as what the food is being consumed in Sri Lankan population. And now coming to the Southeast Asian and the West Asian, that is Indonesia and Turkey. And you know that Indonesians and Turkish people, they do consume significant amount of meat. And we found a very significant interaction between the genetic risk and protein consumption on central obesity. That is, when those individuals consuming the protein consumption, that is animal protein consumption, when the total protein intake exceeds 140 grams per day, that increased the genetic risk of central obesity in Indonesian as well as the Turkish population. A similar finding was also observed in the Brazilian population, that is a South American population, where when the diet was very high in the total protein consumption, that is more than 145 grams of protein per day. And interestingly, in the West African population, that is the Ghanaian population, it was basically the dietary fat consumption. When the total fat increased more than 47 grams of fat per day, or the saturated fats greater than 23.5 grams per day, when that limit was increased, then that was a point when that increased the genetic risk of obesity. So you can see that in different populations, there are different dietary patterns. The differences in the dietary patterns is what is basically the triggering factor for increasing the genetic risk of obesity. So at the moment, we are also doing some studies in the Peruvian population, Nigerian and Malaysian populations as well. So let me share a very interesting finding which we published a few months ago, which was in relation to the adiponectin, circulating adiponectin biomarker. So adiponectin is basically a biomarker of obesity, and high levels of adiponectin is required to prevent the development of obesity. So we found that individuals who had high genetic risk, they had increased risk of cardiometabolic diseases. So this study was done in Asian Indians in the Chennai population, where we looked at 2,000 individuals, where 1,000 individuals had type 2 diabetes and 1,000 individuals were controls, which was basically a case control study. So in this population, people who had high genetic risk, they had increased risk of cardiometabolic diseases. And this was observed after adjusting for all the potential confounders such as age, sex, BMI, and waist circumference. In addition to this, we also identified that people who had high genetic risk, they had significantly lower levels of adiponectin concentration. So if you look at this graph, you can see that individuals who had high genetic risk, that is individuals carrying 2 copies, they had significantly lower levels of adiponectin concentrations. And those who had low levels of adiponectin concentrations, they also had increased risk of cardiometabolic diseases. So now the main question is the association of genetic risk with cardiometabolic diseases, is that being mediated through adiponectin? So in order to test that, we tested for the association of the genetic risk with cardiometabolic diseases by adjusting for adiponectin. That is adjusting for age, sex, BMI, waist circumference, and also adiponectin. But when we adjusted for adiponectin, we did not see any association. You can see that the p-value is not statistically significant. So what this implicates is that the genetic risk with cardiometabolic diseases, the association between the genetic risk and cardiometabolic diseases is dependent on adiponectin concentrations, or in other words, the association of genetic risk with cardiometabolic diseases is being mediated through adiponectin concentrations. So how is this information useful for us? See, for example, if you have a high genetic risk, your adiponectin levels are going to go down. So if your adiponectin levels goes down, then you're going to become genetically predisposed to develop cardiometabolic diseases. So to prevent that, if you know that you have a high genetic risk, one way to prevent the cardiometabolic diseases would be to improve or boost your adiponectin levels through dietary intervention. So how can you boost your adiponectin levels? What are the foods which can boost your adiponectin levels? There are several studies which have clearly highlighted the fact that consuming omega-3 fatty acids can improve your adiponectin concentrations. There is a significant association between omega-3 supplementation and improvement in adiponectin levels. And it is not only the diet, even physical activity can improve your adiponectin levels. So if you're able to improve or boost your adiponectin concentrations through your lifestyle intervention, then that can prevent the development of cardiometabolic diseases. So understanding your genetic risk is very crucial for preventing the development of cardiometabolic diseases. So this is how you translate the findings from nutrigenetics to personalized nutrition. So in summary, genuine collaboration provides the first evidence of all these gene-diet interactions on obesity in these lower middle-income countries. And if you look at implementing the personalized approach, it's very much important that we need to look at the ethnicity of an individual. That is, you cannot use the findings from the Western population to provide a personalized diet for Indians. You need to use the findings from Indian population to provide a personalized diet for Indians. So the ethnicity plays a very important role. The reason is every ethnic group has got a unique DNA. My DNA is different from the DNA of a person who's from Africa. So the DNA is what determines your ethnic group. So you need to use the nutrigenetics findings from that ethnic group to implement personalized diet for that particular population. So just to highlight some of the press release of the findings from the genuine collaboration. So the main take-home message from the genuine collaboration is about like how healthy lifestyle can overcome the genetic risk of heart disease or obesity or even diabetes. A good diet can definitely help Asian Indians to fight against the development of diabetes. So this is about the Adiponectin omega-3 biomarker study, and also we also looking at like various other factors, like how different factors, not just the genetic factor, but also looking at the multi-omics platform, metabolomics or gut microbiome, and how that contributes to the development of obesity. And if you're interested in knowing more about like what I'm doing as part of the genuine collaboration, feel free to go into this publication, which will give you a better understanding of various other studies that we are doing in different parts of the world. So we have seen what nutrigenetics is all about, which is about the gene-diet interaction. Now let's move on to nutrigenomics. So as mentioned before, nutrigenomics refers to the study of the role of nutrients in gene expression. So it all starts with the diet, the food that you're consuming. So the food that you're consuming has got all the nutrients, all the macronutrients and micronutrients, the minerals, amino acids, antioxidants. All these nutrients eventually need to be absorbed by the cell, because you need energy to survive. So these nutrients are being absorbed by the cell, and the nutrients enter into the cell and reaches the cytoplasm. As soon as the nutrients enter into the cytoplasm, there are various molecules which are eagerly waiting for the nutrient to enter into the cytoplasm. And these molecules are nothing but the transcription factors. There are two important functions of the transcription factor. One is basically to bind to the nutrient, and the second important function is to take the nutrient to the nucleus where the DNA is. The nutrient with the transcription factor goes and binds to the DNA, nourishes the DNA, provides energy to the DNA to convert DNA to RNA and RNA to proteins and proteins to metabolites. So in response to a particular nutrient, there are certain RNA molecules which are being produced. And this study of expression of the RNA in response to a nutrient is what you call as transcriptomics. Transcript is nothing but the RNA transcript, and omics means expression, so RNA expression study. And the proteins which are produced in response to a nutrient, and that study is proteomics, and the metabolites which are produced in response to a nutrient, that study is called metabolomics approach. So all these omics approaches, genomics, transcriptomics, proteomics, metabolomics, they all come under one big umbrella, which is a nutrigenomics. And the main purpose of doing these nutrigenomic studies is that if you know that you have an abnormality in your gene expression profile, which is nothing but a reflection of your dietary pattern, so that gives you an opportunity to rectify the issues in the gene expression profile by changing your dietary pattern. So once you adopt a healthy lifestyle or a healthy dietary pattern, that can rectify the problem in the gene expression profile and which can prevent the development of the disease. So these changes in the gene expression profile, they serve as a molecular biomarkers. And understanding these biomarkers will help us to prevent the disease. How can you prevent the disease? By implementing personalized meal plan, that is personalizing your diet based on your gene expression profile. So looking at the gene expression, so this is basically a schematic representation of how the DNA is converted to RNA and RNA to protein. So the transcription and translation are the two important steps in the gene expression pathway. But the gene expression cannot take place on its own. You need that trigger, and which is basically coming from your diet. The foods that you're consuming, they have got all the nutrients, these macronutrients and micronutrients. And these nutrients are basically being absorbed by the cell. They enter into the nucleus where they go and bind to the DNA and regulate the gene expression. So this is what you do, or this is what you study as part of a nutrigenomics approach. So there are different stages in the Nutri Genomics. So one is a genomics, the other one is a transcriptomics, and the other one is a proteomics, and we also have the metabolomics. The genomics, transcriptomics, and proteomics, they all provide the expression of a particular gene or RNA or proteins. And metabolites are very, very crucial because they are the end products of the entire gene expression pathway. And they are the true reflection of your diet. And metabolic profiling is what you call as the metabolomics study. And metabolomics approaches, it's kind of like an upcoming field of area of research, and which is being widely used in order to understand the molecular basis of the health and the disease. So let me give you an overview about like how this metabolic profiling is being carried out to understand the molecular basis of health status and the disease status. So depending on your question, you can either do a human study or an animal study, and you can collect any kind of a sample. And again, that depends on your question, research question. You could do a blood sample, and from blood, you can get the serum sample or the urine sample or fecal sample or even the breast milk. And again, going back to your research question, you can use any kind of a metabolic profiling technology. The widely used one is a mass spec or NMR or any kind of a chromatographic technique. For example, if you're doing a study in 1,000 samples and you're screening the metabolite profile in these 1,000 samples, so which is kind of like a very, which will basically give you a vast amount of data. So that makes it very complicated when it comes down to statistical analysis. The data analysis requires a sophisticated software which can run all these statistical analysis. So I'm not going to go into the technical details, but for your understanding, if you look at the statistics profile, there are different peaks indicating the levels of different metabolites in an individual. So you're basically comparing the cases with control. If you find that there is a high level of certain metabolites in cases compared to controls, which means that the high level of that metabolite that serves as a biomarker and which could also be used as a diagnostic marker for diagnosing the development of certain chronic diseases such as obesity or diabetes or cardiovascular disease. So this is exactly what is being done as part of metabolomics approach. So understanding the metabolites is also equally important because the food that you're consuming, it is not only interacting with your DNA, it is also interacting with your gene expression, with your metabolites. And that area of research is what you call as nutri-metabolomics. The way you have nutrigenetics, you also have the nutri-metabolomics, how a particular nutrient enters inside your body and interacts with the metabolites in your body. So we have seen nutrigenetics, we have seen nutrigenomics, we have seen metabolomics. Now let's have a look at the impact of the diet on gut microbiome. So the gut lumen has got several microorganisms, not just bacteria, but also viruses, phages, and yeast. And we all carry about like nearly 2 kilograms of the microorganisms in our gut. And these microorganisms are very, very important. And we all have a good proportion of the good bacteria and bad bacteria, and they are all important for maintaining a healthy physiological state of an individual. And these microorganisms, they produce certain molecules which are called as the short-chain fatty acids. And these short-chain fatty acids are produced in response to your diet. If you consume a healthy diet, and that can induce the production of the short-chain fatty acid, because these short-chain fatty acids, namely the acetic acid, butyric acid, and propionic acid, they are very much important for maintaining your mental health and your physiological status, and also most importantly, boosting your immunity. So nourishing your DNA is important, and nourishing your gut microbiome is equally important. So let's have a look at the role of gut microbiome in health and disease. So it's kind of a very busy slide, so I'm just going to simplify this for you. So looking at all the unhealthy foods, that is foods which are rich in sugar, or when consuming excessive amount of proteins, or consuming antibiotic frequently, or consuming a lot of junk foods or foods which are rich in saturated fatty acids, so they can all have an impact on the production of the short-chain fatty acids. That is, they can reduce the production of the short-chain fatty acids and increase the production of the TMAO. So TMAO is nothing but trimethylamine oxide. So when you consume foods like such as like the meat or dairy products, so they release this chemical, which is a trimethylamine, so which undergoes oxidation, and that gets converted to trimethylamine N oxide. And this high levels of TMAO has been shown to be significantly associated with increased risk of cardiovascular diseases. And not only that, when the levels of short chain fatty acids decreases, that increases the inflammation of the gut, and that can lead to various chronic diseases such as inflammatory bowel syndrome or even colitis or Crohn's disease. And also it can increase the risk of insulin resistance, decreasing the insulin sensitivity, and thereby leading to the development of type 2 diabetes. On the other hand, if you consume a diet which is rich in fiber-rich foods or consuming probiotics or probiotic supplements, that is basically consuming healthy diet, that can increase the short-chain fatty acid production, improving your antioxidant levels in your body, and which indirectly has got a positive impact on improving your lipid metabolic state and lowering the gut inflammation and improving your insulin sensitivity, and also reducing the risk of infection by boosting your immunity levels. So your gut microbiome is very, very important. And studies have shown that 90% of the type 2 diabetic cases, they seem to have a gut microbiome dysbiosis. So the maintaining a healthy gut microbiome is important for the prevention of the development of obesity, diabetes, and cardiovascular disease. So we have seen the nutrigenetics, nutrigenomics, metabolomics, and also gut microbiome. Now let's move on to the non-genetic area of research, which is the epigenetics. So the genetics is something to deal with the genetic variations, with the changes in the DNA, whereas epigenetics is nothing but a non-genetic research, which means that we are not looking at any changes in the DNA, rather we are looking at the changes in the gene expression. And epigenetics is nothing but how the nutrition is influencing the epigenetic modification. So as mentioned before, the epigenetics is nothing but looking at the heritable changes in gene expression. So where there's no change in the DNA sequence, rather there's a change in the phenotype without a change in the genotype. The phenotype is nothing but the physical representation of an individual, whereas the genotype is a genetic representation of an individual. So there's no change in your genetic makeup, but there is a change in the gene expression, which is influencing your phenotype. And one of the most common epigenetic modification is a DNA methylation, where the cytosine nucleotide is being methylated. That is, when you consume a diet which is rich in methyl donors, so that can enhance the methylation of the cytosine in your DNA. So how DNA methylation regulates the gene expression? For example, if you're not consuming a methyl-rich diet, the methyl-rich diet is considered to be a healthy diet. So if you're not consuming a healthy diet, then which means that your DNA is not methylated. If the DNA is not methylated, then the transcription factors can bind to the DNA and enhances the gene expression. So the unmethylated DNA allows a binding of the transcription factors, which will result in gene expression. But if you consume a healthy diet, such as a methyl-rich diet, the methyl donors, basically the methyl gets added on to the cytosine, and this methylated DNA will prevent the binding of the transcription factors and will prevent the gene expression. So this methylated DNA can result in gene silencing. So gene silencing can control the gene expression because you do not require all the genes to be expressing 24/7. Some of the genes can be switched off when they are not required. You require the expression of the genes only when they are supposed to be expressed. If those genes are expressed throughout the day, then overexpression of the genes can lead to the development of certain diseases such as obesity or diabetes or even cardiovascular disease. So this gene silencing controls the gene expression and preventing you from developing certain diseases. And how can you prevent that? How can you induce gene silencing? By consuming a healthy diet. So the diet is the one that determines the development of the disease. So there's a very strong nutri-epigenetic link between the maternal diet and the fetal growth. And that's why we say the first 1000 days of a child's life is very, very crucial. Several studies have clearly shown that the unbalanced maternal diet during pregnancy have shown to have an impact on the fetal growth. And when these kids, when they become adults, that has increased the cardiovascular disease risk in adulthood. And also the seasonal variations in maternal methyl donor nutrient intake during the periconceptional period has been shown to influence the plasma biomarkers. And this has got an impact on changes in the methylation of certain genes. And maternal obesity, that is obesity during pregnancy, or even diabetes during pregnancy, which is what we call as the gestational diabetes, they can all have a significant impact on DNA methylation changes. And also micronutrient deficiency during pregnancy can influence the DNA methylation. So let me briefly touch upon the micronutrient deficiency during pregnancy, so which I'd already mentioned before in one of the previous slides, where I was talking about the vitamin D pregnant mother cohort study. So this is a study that we did in Indonesian women. So where we followed up the pregnant mothers right from trimester 1, 2, and 3, and we also measured the anthropometric, the newborn anthropometric measurements. So in the first part of the study, we were able to show that the pregnant mothers who had high genetic risk, they had significantly lower levels of 25-hydroxyvitamin D concentrations. So this association was observed in the third trimester. Now let's have a look at the babies that were given birth by these pregnant mothers who had high genetic risk. Interestingly, we found that these pregnant mothers who had high genetic risk and those who had vitamin D deficiency, they gave birth to babies which had a small head circumference that is less than 35 centimeters. So which is a clear indication of the fact that the micronutrient deficiency during pregnancy can have a significant impact on the fetal growth. And small head circumference, that is, several prospective studies have clearly identified a significant correlation between the small head circumference for the babies at birth in relation to the development of cardiovascular diseases when these kids become adults. So which is a clear indication of the fact that maintaining a healthy nutrient state is very much important during pregnancy to prevent abnormalities in relation to the fetal growth. So foods rich in methyl donors are very, very important. That's why like what you do and what you eat in the first 1000 days of your life can make a significant difference for the rest of your life. So we have seen the different aspects of different stages, like starting right from nutrigenetics, nutrigenomics, metabolomics, gut microbiome, and epigenetics. Now, in terms of translating nutrigenetics to precision nutrition, the food that you're consuming, they enter inside your body into 4 different compartments, starting with interacting with your DNA, and that is what we call as the gene-diet interaction or the nutrigenetics area of research. And it goes into another compartment where it interacts with the metabolites. And that area of research is what we call as the Nutri-Metabolomics. And the food also enters into the gut lumen, where it interacts with the gut microorganisms. And that is what you call as the gut microbial profiling and metagenomics. And last but not least, it also interacts with your DNA, that is having an impact on the gene expression by modifying the epigenetic modifications, such as the DNA methylation. So all these 4 compartments are functioning in parallel as soon as you consume any particular food. So if you need to implement precision nutrition or the personalized nutrition, it is important that you need to look at it as a 4-way interaction, which is the metagenome, hyperbolome, epigenome diet interaction. And understanding this interaction is crucial for the implementation of the precision nutrition. But the key question over here is, how are we going to integrate these 4 compartments together to develop a personalized diet? How are we going to put everything together? We need an integrated platform to bring about a revolution in the field of human nutrition. So how are we going to do that? For which we will need the machine learning approaches, or in other words, the artificial intelligence platform. So we need to combine the multi-omics approach with that of the dietary intake, like taking into account of the nutrigenetics, nutrigenomics, gut microbiome, your physical activity levels, your dietary intake, your metabolite levels, your anthropometric measurements, your clinical biochemical measurements. So we need to put everything together and we need to do a data analysis. But this can be done in two different ways. One is basically using the traditional statistical approaches, which is kind of like very sophisticated and time consuming. But the other easier way, well, easier in the sense like it's much more easier in terms of feeding in the information which will run the program for you to get the personalized diet as an output. So which is basically a machine learning approach where you're giving different data sets or the different multi-omics approaches, the data from various multi-omics approaches, the dietary intake and the lifestyle factors together, and you can use any kind of an approach you have, like the supervised learning or the unsupervised learning, machine learning approach to implement the precision nutrition approach. These are all ongoing research activities which are happening at the moment. So the key question is, would you let your phone tell you what to eat? For example, if you have an app which takes into account of your gene, your metabolites, your epigenetics, your gut microbiome, and also your dietary intake, and will you let your phone tell you what to eat? So that is the most important question. So which is basically an online personalized nutrition advice, and also an app that can assess your dietary intake and which can propose valid personalized nutrition advice for adults. There are various apps which are available. That is, the personalized nutrition apps which are available, and they can be accessed on a range of devices. So the basic idea is basically to enable more people to benefit from tailored dietary advice. But all these apps are taking into consideration of only the genetic factor. They do not provide any information in relation to your metabolomics or your gut microbiome or even your epigenetics. So that is a long way before we could reach that level of implementing precision nutrition. So a lot of research is being carried out. So looking at the precision nutrition in the era of artificial intelligence, so we need to take into consideration of all the factors. That is, this side of the figure, like we had already looked at the genetic, epigenetic, microbiome, metabolomics, proteomics, your nutrition status and physical activity. But on this side, you can see that there are various other factors like your stress levels, your sleep patterns, your age, your geographical condition, your socioeconomic status, your early life, what have you been exposed to? Have you been exposed to certain chemicals? And also looking at various other lifestyle factors like your smoking patterns or your consumption of the tobacco. So various deep phenotyping is required to understand and implement precision nutrition. And how are we going to integrate all this information? Of course, as I said before, we definitely need to carry out a big data analysis, integrating and developing machine learning approaches, or in other words, artificial intelligence powered decision making, because this is exactly the one that is going to implement the strategies for personalized nutrition approaches, which can eventually prevent the development of the chronic diseases. But at the moment, we are still like carrying out various research activities of how we are going to integrate everything together, and we still do not have a personalized nutrition approach which takes into consideration of all these factors together. So we are very, very far. Like it will take at least another decade or at least 2 decades to reach that level of implementing the precision nutrition approach. So on this note, I would like to thank all my genuine collaborators, and thanks to all the DCardia collaborators. And finally, thanks to the organizing committee once again for inviting me. And also thank you all so much for listening to my talk. If you have any questions, you can drop me an email at v.karani@reading.ac.uk. And I'm available on Facebook, LinkedIn, Instagram, and Twitter as Vimal Karani. So thank you so much. And I'm happy to take any questions.
Thank you, sir. It was a great pleasure to hear from you. Trina, we have questions. You can put the question to sir.
I guess there is not many questions because you have already explained all the concepts very simply and clearly. But I have some questions from my side. So can I ask you that?
Yeah, sure. Please do.
Yes. My first question is, what is the mechanism that relates adiponectin and omega-3 fatty acids that you mentioned in your slide?
Okay, yeah, that's a very good question. So these omega-3 fatty acids are basically those nutrients like which basically goes and binds to this molecule which is called as the peroxisome proliferated activated receptor. So the PPA gamma basically activates the adipogenesis process. So this PPA gamma requires an kind of like a trigger, and that trigger is being given by the omega-3 fatty acids. So these fatty acids can go and bind to the PPA gamma, and this PPA gamma can go and bind to the genes which are being positively regulated. And one of these genes is basically the adiponectin gene. So this PPA gamma can go and bind to this adiponectin gene by forming an heterodimer with the PGC1 alpha, which is a transcriptional coactivator. So they interact and go and bind to adiponectin and regulate the adiponectin expression. That leads to the production of the adiponectin in the body. Did I answer your question?
Yes, sir. Yes, sir. Absolutely. Thank you, sir. Sir, I have two more questions for you.
Yeah,
And
sure.
the second one is, what is the general methods to understand one's gene expression or his vulnerability or risk percentage to certain diseases?
Sorry, I didn't get your question. So what methods for gene expression?
Any methods or easy methods to understand one's gene expression or understand one gene expression or their vulnerability or risk percentage to certain metabolic diseases? How can they figure out that?
Well, majority of the gene expression profiles or data that is available is mainly from the animal studies. But there are some studies that have been carried out in humans as well, but they have been quite inconsistent. So that's exactly why I said like we are very, very far from implementing such personalized or precision nutrition approaches, taking into consideration of all these genomics and proteomics. But what gene testing centers are doing at the moment, like we have several gene testing centers across the world, and even in India we have like several gene testing centers, and what they're doing is they're just looking at the genetic variations in the DNA, and they're trying to see whether you carry 0 copy or 1 copy or 2 copies, and depending upon the level of genetic risk, they're providing you a personalized diet. But we haven't reached the level of where you can look at your gene expression profile and identify the risk, because we still do not have a strong, valid evidence that gene expression profile abnormalities can lead to cardiometabolic diseases. Because some studies have shown a significant association, and some studies have failed to demonstrate such associations.
Okay, sir. And the last one is it possible to rectify the effects of mother's diet on only by modifying the diet during pregnancy, or there is any effect of diet that the mother is consuming or consumed her whole life?
Well, I mean, of course, as I said, like the first 1,000 days of a child's life is very, very important, because that is the time like when the genomic imprinting takes place as well. And of course, the mother, when she was a child, I mean, that diet during the first 1,000 days also plays an important role. But of course, during pregnancy and just before pregnancy is also a very crucial period, like for the future generations, like, I mean, having an impact on the fetal growth. For example, like the vitamin B12 supplementation or folate supplementation is being mandatory, especially for pregnant mothers. But whereas like there are several other micronutrients, several vitamins, such as vitamin D. So not many people are looking at the vitamin D deficiency. And that is not at the moment a mandatory supplementation for pregnant mothers. If they are insufficient, then they can go for vitamin D supplementation. But not all pregnant mothers are basically testing for the vitamin D deficiency level during pregnancy, because that's not being considered as an important vitamin supplementation. So I would suggest that, like, I mean, they need to screen all the vitamin D markers, or not all the vitamin markers, not just B12, but also vitamin E, and also looking at the vitamin D and looking at how, if they are vitamin D insufficient or vitamin D deficient, then they should definitely go for the supplementation, which can prevent abnormalities in the fetal growth.
Okay, sir. Thank you, sir. Sir, there is another question from our Dean Madam.
Yeah.
The question is, according to you, how diet can affect the SNP of various genes?
Well, the diet is not affecting the SNP. What is happening is you cannot change your DNA based on your diet. the genetic variation. For example, if you are born with low genetic risk or moderate genetic risk or high genetic risk, you have to live with that level of genetic risk until the last day of your life. You cannot change your genetic risk. But what can change your genetic risk is basically your diet. Your diet can overcome the genetic risk, or your diet can increase your genetic risk. If you're consuming a healthy diet, that can overcome your genetic risk. But if you're consuming an unhealthy diet, that's going to increase your genetic risk. So the level of genetic risk can be changed depending upon your dietary pattern. But what cannot be changed is the changes in the DNA.
Yes, sir. Sir, there is another one from her also. And
Yeah.
the question is, beside omic approaches, how physiological factors can be assessed in various epigenetic approaches?
Besides the, sorry, come again. Besides the,
I am repeating the question.
yeah.
Besides the omic approaches, how physiological factors can be assessed in various epigenetic approaches?
Okay, yeah. So epigenetics is kind of a non-genetic area of research, whereas the omics, the genetics and genomics, they are completely different area of research. They are completely two different ways. So one way to look at your physiological state is looking at the multi-omics approaches, looking at the gene expression profile or the transcriptome profile or your proteome profile or metabolite profile. And in terms of the epigenetic markers, you can look at the DNA methylation or histone modification. So these are all basically controlling your gene expression. But coming to the physiological factors, there are various other external factors or environmental stimuli which can influence your physiological state, like your sleep patterns or your physical activity and your diet, and various other exposures can have an influence. There are various methods by which you can assess your stress levels, your sleep patterns, and various other factors as well.
That's all, sir. Thank you, sir, for your such an enriching, wonderful session. We are overwhelmed to have you with us. Thank you so much, sir.
Thank you. Thank you very much for inviting me and for organizing this event as part of the National Nutrition Month. And thank you all very much once again.
Thank you, sir.
Thank you. Thank you very much.
Do we have our second speaker?
I think that there is another half an hour left before the second session is scheduled for. So I would request you to kindly take decision what should we do before our second speaker joins.
Purbadi?
This was a very
Actually, I
enlightening
think
lecture, and I think that yes, we can discuss. And actually, this our next speaker is maybe joining a little bit later, and it would be very nice to just discuss something that is it is not only this international webinar series that our department is going to organize. We have actually things which are lined up for the entire month, nutrition month, and there is this slogan competition, and for those students who are viewing today's lecture series are there present, it is an earnest request. Please participate in the activities like poster writing competition. There are various recipe competitions where we will be judging the best recipes, 3 or 4 recipes. And next there is a kind of a skit kind of a thing. Students are being encouraged to make certain small skits. The pro forma has already been circulated. So let us all join hands and make this celebration a very successful one. What about Kubalidi, our next speaker?
Just checking.
So please bear with us for a few moments.
No, he hasn't joined yet. He hasn't joined yet.
We'll
I'm
take
checking.
a small break and then we will continue with the deliberations.
I think we can take a small break, 10 minutes break, and then we can join back, isn't it?
Yeah, that would be nice.
Then Pubalidi, can you tell us? Shall we take a break of 10 minutes and then join back? Diksha, can you stop sharing your presentation? I'll be presenting a video, short video on the food from waste. So,
Yes,
mom,
ma'am.
just tell, can you see the video? I'm sharing it
Okay,
now.
ma'am.
Huh? It's coming.
Yes, ma'am.
Volume is there. Just see.
Not clear, ma'am.
Can you hear the volume?
No, actually, video volume, that is the most important.
Can you hear now?
No.
Ma'am, you have muted yourself everywhere. Yeah, now little bit coming.
Can you hear the volume?
It's very low, ma'am.
Professor Weber has already joined. Anna, Professor Weber has joined.
Okay.
Welcome, Professor Weber. Thank you for accepting our invitation. It's wonderful to have you in this session.
Well, thank you very much for the invitation. It's my pleasure to meet you all today and to talk to you today.
Actually, I'm a professor of nutrition, and I teach space nutrition. So I'm very much interested, and and one of my student, he searched for you, and ultimately you accepted our invitation. Thank you, sir. Thank you.
Yeah,
very
thank you very much for the student. I think his name is Niloy. So he
Yes.
contacted me, tried to catch me several times, and now I'm here. So thank you for being very persistent, Niloy, to make this happen. Yeah, again, it's my pleasure to be invited and to talk to you beautiful people in India about human spaceflight. My personal area of science is not really nutrition. I know a little bit of nutrition, of course, but my specialization is actually the musculoskeletal system and exercise
I'm a
physiology.
physiologist. I teach nutrition, but I'm a physiologist.
Okay, great. Yeah, my background is space physiology. I had received a PhD degree in 2013, and it was based on space physiology.
Thank you, sir. Thank you very much.
We welcome you, sir, for today's session. Trina, give sir an introduction.
Yes, ma'am. Now we'll move on to the next session. Our next speaker is Dr. Tobias Weber. Tobias Weber is currently working as researcher in European Space Agency, Paris, France, and KBR Weil, Cologne, Germany. He is also a part of space medicine team at the European Astronaut Center in Cologne, Germany. Dr. Tobias Weber obtained his PhD degree in space physiology in 2013 in collaboration with the German Sports University and the German Aerospace Center, and then completed his postdoctorate from European Space Agency. His research focuses on various aspects of space physiology and on projects trying to optimize healthcare for astronauts pre, in, and post flight, with the aim to also make his research findings and technology developments applicable for the terrestrial population. Dr. Weber is also a licensed strength and conditioning coach. He has 31 research articles published in some of the most renowned peer-reviewed journals, and he has also contributed as editor and reviewer in many international publications. He will be speaking on the topic entitled Musculoskeletal Deconditioning in Space Etiology and Non-Pharmaceutical Countermeasure Concept. I would request all the participants to turn off their audio and video during the lecture session and put their questions in the chat box. Welcome, sir. The platform is yours now.
Thank you very much for the kind words and the introduction. Let me quickly close my window because we are here next to the airport and I want to avoid any background noise. So yeah, thank you very much for the introduction. Probably the best will be that I start with my 30-minute presentation, and then I have plenty of time to answer your questions in real time, if that's possible.
Yes, sir, of course.
Okay, so let me try to share my screen. Okay.
I want to hear a little more, if possible.
Okay, I think you should be able to see my screen now. Can you?
Yes.
Can you confirm that? Okay.
Yes.
Yeah, so again, I will start with, I mean, I'm not really a nutritional expert, so I thought for you guys, it would make more sense if I talk a little bit more generic about the problems that we see with our crew in space. And the topic of my lecture is musculoskeletal deconditioning in space. I will talk a little bit about the etiology. So why does it occur? Why do we have these problems? And about what we do about it. So about non-pharmaceutical countermeasure concepts that we apply to our astronauts, and that and also about a few countermeasures that we might apply in the future, so that we are currently researching. And of course, when we talk about astronauts, there's always a big overlap with terrestrial pathologies that we see. For example, people who are exposed to long periods in bed, they kind of experience the same physiological deconditioning that astronauts experience in space, because they have no gravity, hence they don't use their muscles anymore, they have no loading on the skeletal system, they degenerate cardiovascularly. So there's a lot in common. So I think it's quite a nice model to study astronauts, but I think there are also many benefits to learn how to improve reconditioning and prevention for terrestrial patients who are lying in bed for longer periods because of certain diseases and pathologies. So let me first introduce you to our center. So I'm actually based in Germany, in Cologne. I don't know if you all know where Cologne is. Germany is in the middle of Europe, and Cologne is kind of in the western part of Germany. I think for Indian conditions, it's rather a small city. We have 1 million citizens, but we have the home base of all European astronauts here in Cologne. So I'm located at the European Astronaut Center in Cologne, which is as I said, the home base for all European astronauts. We select the astronauts here, we train them here, and I work in the space medicine team that takes care of all medical aspects before, after, and during space missions of the European crew. So within the European Astronaut Center, it's a rather small center. We have 100 experts. Of course, we have the astronaut corps themselves. We have currently 8 active European astronauts. Maybe you've seen in the media that ESA has just announced a new call for astronaut recruitment, and we've had more than 20,000 applications. So there will be a new generation of astronauts coming in the near future. So we have, as I said, we have the astronaut corps itself. Then we have a astronaut training division that takes care of astronaut training, basic training. So that's a 2-year phase where they learn everything that they need to learn about being an astronaut. And they also take care of mission-specific training. So during their training, during their stay in space, astronauts have to perform science. And our astronaut trainers, they teach them how to perform science experiments, because as you can imagine, an astronaut has to do more than, on average, 150 different science experiments. And you need to have some proper training in order to do them in a way to produce reliable and useful results. And then we have medical operations. That is kind of my team. I work in the space medicine team. We have astronaut support, an astronaut support team to help them about everything that is needed when they prepare for their missions, to take care of their families, to help them traveling, to generally take care of them. We have crew operation tasks. So we have people who are in direct contact with the crew. So we have an operation center here. We have people sitting on the console who are in direct contact with the International Space Station to help them through the day and to also provide some guidance. And of course, every time you see ESA astronauts in public media, be it on social media, Instagram, Facebook, public TV, educational TV, this is all coordinated through our colleagues from the public relations team. So I have already talked about the International Space Station. I don't know if you're all familiar with the International Space Station, but this has been a big station in low low Earth orbit that is orbiting the Earth in approximately 400 kilometers. So it takes approximately 90 minutes for one orbit around the Earth. And this station exists since more than 20 years now. So it's a big European, or no, not European, big international research, microgravity research laboratory. However, the time of the ISS will sooner or later come to an end, probably towards the end of this decade. And we are now on a simultaneously working on another program, which is called the Lunar Gateway. You can see a small or little sketch of the Lunar Gateway here on the right-hand side. It's going to be a significantly smaller station than the ISS, but the difference is, of course, that it's going to be in deep space. And I don't know if you can see the video on the left-hand side, if it really plays. Can you confirm that you can see that it's running, the video?
Yes, sir. The video is running.
Okay. So what you see in the video is the orbit. So you can see the Earth in the middle, and then you can see the Moon orbiting the Earth. Of course, we all know that it does that since billions of years. And then we see the orbit of the gateway. So it's kind of a very elliptical orbit that takes approximately 10 days. And from the Lunar Gateway, it is planned to have the first lunar surface explorations since the Apollo missions. And we hope that we can see the first human beings back on the lunar surface even in this decade. So both the ISS and the Gateway program run in parallel for the next couple of years. But sooner or later, the ISS will very likely retire. And then the only human outpost for Europeans will then be the Lunar Gateway, like a deep space station orbiting the Moon. And the key differences when we compare that to the international space stations are that the isolation there is more severe, because as you can imagine, now when the ISS is only 400 kilometers away, we can evacuate the ISS relatively quickly. So if there's a fire on board, or if there's some sort of medical emergency, we can bring our astronauts down within 24 hours to the surface of the Earth, and to guarantee that they are in medical care. And also because we are further away of the Earth and the atmosphere and the Earth magnetic field, the radiation exposure in the Lunar Gateway will be much higher than currently on the ISS. We have longer evacuation times, and of course, if an astronaut after a certain period in microgravity has to do a lunar surface exploration to explore the lunar surface, to maybe build something there's nobody there awaiting them. That's also a big difference compared to the ISS, because now when astronauts land after let's say 6 months on the ISS, there's direct medical care. So they used to land in Kazakhstan when they launched with the Russians. Now that the Americans have also their own launcher program again, they land in the Pacific, but they directly have full attention, and a whole rescue team will be waiting there for them to take care of them. And all this is of course going to be different when we have astronauts walking on the Moon again. So when we are in space, basically the whole human body adapts to the different environment. The main reason is of course the absence of gravity. We have phenomena from head to body, and basically all physiological systems are affected by the absence of gravity. For the talk today, for the lecture today, I would like to focus on weight-bearing bones and muscle and weight-bearing muscles. because this is kind of my specialization, and I think I know most about this phenomenon. But I think the take-home message for you is that all physiological systems will adapt to microgravity, and we see some adaptations that are unfortunate because we consider them pathological adaptations, and some adaptations are just normal, and they go back to normal as soon as the astronauts get back to Earth. So when we look at the data of the skeletal system, also skeletal adaptation, microgravity, we can see on the left-hand side, you can see a graph. It's expressed in percentage, the bone loss that occurs after a 6-month space mission. And you can see that it's basically, non-surprisingly, weight-bearing bones that help us in 1G to counteract the effects of gravity, that lose the most bone mineral. The old saying, what we use in exercise physiology, is lose it or use it, and this is a very good example of that phenomenon. So the bones, especially the femur, the tibia, the pelvis, but also the lumbar spine, they are heavily affected by the absence of gravity. So the body reacts in a way to resorb bone mineral content, because the body thinks a microgravity is no longer needed. But what you also see, and that's also similar to what we see on Earth, is that we have a high inter-individual variability. So not all astronauts react in the same way. So we see always that the mean is, so we have always a negative effect if you only look at the mean, but we also have some outliers who actually seem to not lose bone, or to sometimes even gain bone, which is kind of surprising, but it's kind of the human condition, that we are all the same, yet we are all different. How we react to different environments, how we react to different drugs, how we react to different interventions. So as a scientist, I find it always important to look at the mean changes of a certain intervention, but also not forget about the individuals, because especially if you have small sample sizes, which is the case for astronauts, then it's very important to understand why certain people react in a certain way. And the etiology of bone loss in microgravity is fairly simple. Back in 1987, there was a famous researcher called Harold Frost, and he introduced the concept of the Mechanostat Theory. That means that the bones have a kind of control mechanism. You will always experience bone loss if the mechanical load, and then hence the strain rate on a bone, is too low, like in bed rest, for example, or like in microgravity, or if you lead a very sedentary lifestyle without lots of physical activity. Then he called a state an adapted state, where you don't lose bone, or where bone resorption and bone formation are kind of in an equilibrium. So you have neither bone loss, but you have also no bone accrual. And then there's the state of bone gain, kind of overload. That is when you have, for example, when you do intense sports or weightlifting kind of exercises, sprint training, football players, tennis players, they have very high mechanical loads on their bones, and they tend to gain bone on the relevant sites. So this just very broad overview is called Mechanostat Theory, and it was introduced by the researcher Harold Frost in the 1980s. So, of course, in space, as I said before, we don't have gravity, or we call it microgravity. Of course, gravity is always there. But since the station is orbiting, it's kind of a free fall that the astronauts experienced. It's like a permanent free fall, and that leads to microgravity. That means they don't experience 1G as we do on Earth. So they float through the station, and that's what we see when we see astronauts in space, floating through the air as if there was no gravity. And of course, this leads to a significant reduction of mechanical forces acting on the bone. So compression will be significantly reduced. They don't need to walk anymore, so there will be less shear forces acting on the bone. And this affects mostly, of course, the weight-bearing bones. So the bones that we use on Earth to walk, to run, to stand upright, to even sit, like me, upright with the lower spine being loaded through gravity, and all this is not there in gravity. So this is the main reason for the bone loss that we see in microgravity. And of course, the underlying effect, what bone needs actually to stay healthy, is bone strain. And bone strain is just a unitless expression of how much bone is deformed. It can be compressed, there can be torsion, it can be bent, but we know that a certain strain rate is really needed to maintain skeletal integrity and health of the bone. And very impressive is there are some very impressive studies from, for example, tennis players. This is not actually a scan of Roger Federer, but it shows it's from a professional tennis player, and you can see that in the dominant arm, the bone, the radius and the arms of the forearm are much thicker, much stronger in the dominant arm compared to the non-dominant arm. And of course, the main reason for that is that they play on a daily basis with a racket, and they have very high forces, very many repetitions every day for long periods of time. So bones adapt and get stronger. And when we come to muscular adaptation to microgravity, the story is very similar. Here on the left-hand side, you can see two figures of the calf muscle, the gastrocnemius medialis, the soleus muscles. So two major calf muscles, and you can see that there's also a significant loss of volume and strength in these muscles, it's a very similar story to the bones. And again, the kind of catchy phrase applies here as well: use it or lose it. So in space, astronauts are not necessarily required to lose their calf muscles. So because they don't need to walk, they don't need to run, they just float through the station. And accordingly, the gastrocnemius medialis muscle and the soleus muscles, as one of the main plantar flexor muscles, calf muscles, they will atrophy, and they will get weaker and smaller. And the same is true for paraspinal muscles, for pelvic muscles. So basically all the muscles that are responsible to keep us upright and to help us to walk and run in 1G. So they tend to atrophy in microgravity. The etiology of muscle atrophy in microgravity is also very similar to the etiology of bone. It's very complex, actually, how muscle tends to atrophy and hypertrophy. But I think the key here is a term called mechanotransduction. So muscle tissue, muscle cells, they need to be they need to be mechanically triggered. And we usually achieve that if you go to the gym and you lift weights. There's a certain mechanical stimulus that leads to a very complex downward cascade of signaling pathways that ultimately lead to hypertrophy. So your muscle gets stronger and bigger. On the other side, if you don't have mechanical stimuli, if you don't use your muscles, then the signaling pathway works in a different way. So you have more muscle atrophy. And that's the case, for example, in bed rest and disuse. If you have, for example, a broken arm and you have to put it in a cast for six weeks, you know that after 6 weeks, your muscle will be weaker. And atrophy, of course, occurs also in aging and in microgravity. Whilst hypertrophy usually occurs when you exercise or when you're growing. So when you go through childhood, teenagehood until you're an adult, you experience hypertrophy. But again, a very key stimulus is mechanical stimulation, and the term mechanotransduction. In simplified terms, mechanotransduction means that a mechanical signal is translated into electric and biochemical signals that act on a cellular level to promote either hypertrophy, so muscle growth, or atrophy on the other side, which means muscle loss. Of course, we don't simply accept that fact, and it would actually be unethical if we would do that. So if you imagine astronauts now, the average time spent on the ISS is approximately 6 months. So 6 months in microgravity. If we wouldn't do any exercise on board the station, then our astronauts would come back in a very miserable shape. So they would be completely deconditioned, and it would take very long to bring them back to shape, and probably we would even cause some long-term damage to them. So that's why we sometimes we have to force them, but actually most of the astronauts, they like to exercise on the ISS. And from the left to right, you can see the 3 currently used exercise devices that we have on the ISS. The first one is called the ARED. ARED stands for Advanced Resistive Exercise Device. This is basically a multi-use resistive exercise device where you can train with high loads to do squats, for example, bench presses, to train your muscles and to keep fit. And then in the middle picture, you can see a CVIS. CVIS is just an abbreviation, but it's basically a cycle ergometer where astronauts can bike to train their cardiovascular fitness. And on the right-hand side, you can see a T2 treadmill, where the astronauts strap themselves down with bungee cords, where they run. And running, of course, is a twofold exercise. You have the impacts and you have the mechanical stimulation of the weight-bearing bones, but simultaneously, of course, train the cardiovascular system as well. And we have a very, now we are pretty successful to bring astronauts back after 6 months in space in reasonable shape. They still need to undergo 2-week recovery. But if you look at the footage from NASA and ESA crew members coming back after 6 months in space, they are actually in a pretty good shape. And one reason is because we have always tried to improve our countermeasures, the exercise countermeasures. And they have to train on a daily basis for 90 minutes approximately. So that's how they, kind of counteract the effects of microgravity, mainly through exercise. That's why exercise is really a key feature of our countermeasure program. We have, of course, some other countermeasures that are currently under consideration that we that are tested in ground-based studies. So most of them have not been tested on the ISS, but I just wanted to introduce them here because some of it might be interesting in the future for astronauts, and certainly most of it is interesting for patients on the ground. So we do a lot of testing with short arm centrifuges. The principle of centrifugation is very simple. You have a centrifuge, and because of the inertia, you kind of artificially create a gravity gradient, so that at their feet, astronauts, but it hasn't flown yet, but the participants who lie on the centrifuge experience approximately 1 point, depending on the rotation speed, of course, but they can experience up to 2G at the foot level. Sorry. So centrifugation would be one option to artificially generate gravity in space. That's why we call it artificial gravity. Then another thing, which I personally find very interesting, is motor imagery training. You probably know this from high performance athletes when you look at skiers, for example, or bobsledders, or even football players. They use that, or probably in India, cricket players do that as well. They mentally prepare and they go through certain movements that they perform, and this kind of research comes from spinal cord injuries and from stroke patients, and it's very interesting and very promising that the fact that you have so much mental power in your brain when you think about a certain movement, that certain regions in your brain are getting activated, similar to as if you would actually do the movement. And of course, we're also interested to learn more about this. For example, if you imagine that someone is on a mission to the Mars and would be for 250 days in an ideal scenario in a very small capsule, you probably, if we wouldn't, and in a capsule, we probably wouldn't have a treadmill. So we would need to find a way to train that person to not unlearn how to walk, because again, if you arrive on the surface of the Mars, there's nobody to expect you there. So we directly need fully functioning astronauts in order to assure that they don't injure themselves, or that they don't have a fatal accident on the surface of Mars. So I find this topic of motor imagery very fascinating. And then on the right hand side, you can see a picture of a resistive vibration exercise device. I don't know how the situation is in India, but in Europe, you can see many studios now all over the place. They promote resistive vibration platforms where you stand and you basically get shaken by that platform. And the principle there is also very simple. You know, they want to promote a mechanical stimulus to the musculoskeletal system in order to maintain musculoskeletal integrity. This has also been tested in bed rest studies. We in the spaceflight environment, we use bed rest as one of the most common ground-based analogs to mimic or to simulate the effects of actual microgravity. And there were some very positive results of the resistive vibration exercise. The problem, though, with resistive vibration, if you imagine that being applied on a space station, you always have the problem of vibration. And vibration in space on a small station doesn't make our engineers very happy. So I'm a little bit skeptical if this will ever be applied in actual microgravity, so in space flight. But of course, there are certain applications in terrestrial medicine, terrestrial rehabilitation, where this might be a useful tool. Another exercise countermeasure that we have been studying for a while now is called blood flow restriction training. The principle is also very simple. So you restrict your blood flow while you are training for a certain period. It's not very pleasant, to be honest. I've tried it myself. It's kind of painful, but studies have shown that you have more muscle growth in the area where you applied the blood flow restriction. And the reason for that is that when you do that, you restrict the blood flow. That means you have more hypoxia in the tissue, and you have the metabolites of muscle work staying longer in your tissue. And we know that most of these metabolites are a hypertrophy trigger. So they lead to a signaling cascade that leads to muscle growth. But of course, again, the downside here is that it's not a very pleasant form of training. It can be painful, and there are also some risks associated with it. So not everybody can do this. Then on the bottom, you see a picture of a guy lying in a so-called lower body negative pressure device. This is something to basically train the cardiovascular system. So one problem that we see with astronauts when they come back from space is that they experience something that we call autostatic intolerance. So basically they tend to faint. And the reason for that is that they lose plasma in space, and the baroreceptors that maintain our brain perfusion in 1G on Earth, they kind of misfunction when upon the transition from microgravity to 1G. And that leads, especially in the old days, that led to astronauts when they stood up for the first time, they got dizzy and they fainted. Some of you might know this phenomenon when you lie on the sofa for a longer time and then you stand up and you are a little bit dizzy, and it takes some time before your blood pressure and your heart rate adapt to maintain the brain perfusion. And in spaceflight, we have used, and we hopefully will use a device again that simulates gravitational stress by pulling, so creating a vacuum from your waist downwards to pull the blood towards your legs to train your cardiovascular system to kind of fight against that, which is kind of similar to what we do on Earth every time we stand up from a chair, for example, or every morning we wake up from our beds and walk. And then on the last picture shows something that you're also probably familiar with, similar to resistive vibration exercise. In Europe, you can see many studios that promote this kind of exercise now. Now it's called electromyostimulation. The principle here is also very simple. You have electrodes above superficial muscles. There's a certain currents going through the electrodes and the muscle will contract. That's one way to make a muscle heavily contract without the use of actual weights or actual machine. So this is also an interesting topic, and we will now have the first crew member who is launching this autumn, who will have such a suit, exactly this suit, and who will test it in space. And then, of course, I have to talk about the most promising countermeasure thus far, and this is plyometrics. Plyometrics, I don't know if you're familiar with the principle of plyometrics, but plyometrics means basically that you have very high forces for a short amount of time, like, for example, when you do rope skipping or when you do jumping, with a very quick and short stretch reflex cycle. So you have your muscle contract and being so concentric and eccentric phase are very fast. So you have a high rate of force development. And we had a bed rest study for 60 days where people just trained for 5 days per day, where they did some very aggressive jumps in that supine jump sled here. So they did counter movement jumps and ankle hops. And you can see on the top graph here, when you look at the bone mineral density, and this is very remarkable, you can see that the control group, they lost bone. So the control group didn't do anything. But the group that jumped, they didn't lose bone at all after 60 days. And this was only a 5-day training intervention per day. And 5-minute training intervention per day. And the rest of the day, the participants just lay in bed. So that's very remarkable. And you could see that for the tibia, on different sides, bone mineral density and bone mineral content were maintained. And the same applies to body composition. So if you look at the dark or the solid black curve, which is the jump group, when you look at the body composition, so body mass was reduced. That's a normal phenomenon that they go down because blood plasma is reduced. But when you look at the fat mass, you can see that there's a significant difference between the group that didn't jump and the black group that jumped. And the same applies to lean mass. So lean mass is basically muscle mass. So in the control group that didn't do the jump intervention, we saw a significant reduction of muscle mass, whilst the group that did a 5-minute jumping intervention every day, they almost managed to maintain the initial level of lean mass. And this is also reflected in muscle strength. So also very remarkable. So knee extension and plantar flexion, knee flexion and plantar flexion. So ankle flexion and knee flexion and knee extension and ankle flexion. You could see that in the jump group, again, the dark solid line, the participants were able to maintain their muscle strength after 60 days, which is, again, very remarkable to me. So we can say, based on this, plyometrics or jumping is probably one of the most promising countermeasures that we currently have. And this is relevant for astronauts, but it could also be relevant for people lying in bed for prolonged periods. And yeah, the question is now, why hopping? Why is hopping so efficient? And the explanation for that is actually relatively simple when you look at the biomechanics or the mechanics behind that. So as I said before, bones need mechanical stimuli. So they need to have a certain force that is applied on the bone. So when you look when you look at how the force at the tibia is generated, it's a sum of the muscle force that is acting on the Achilles tendon. Here now the example of the tibia, and the gravity force. So the force of the mass and the acceleration. And if we look at the body mass, of course, when you're standing and hopping, it's similar. It's, let's say you're a person, you weigh 70 kilograms, you stand, the body mass is the same. The ground reaction force, though, when you're hopping is different. When you're standing, the ground reaction force, so based on Newton's law, is 700 Newton. When you're hopping, it's already 2,500 Newtons. The acceleration here, and that's the reason for the increase is the acceleration, because when you're standing, you have 1g Earth acceleration. When you're hopping, you have 3.5g Earth. You have 3.5g acceleration, and the reason for the significantly increased muscular forces when you're hopping or when you're walking is the mechanical advantage. When you look at the right-hand side again, you can see that from the point of force application to the fulcrum, so where the ankle sits, compared to the point of force application of the Achilles tendon, together with the calf muscles, you have 1 to 3 mechanical advantage, or one could also say disadvantage. This means that either every force that is generated at the point of force application here needs to be compensated by the calf muscle. And the calf muscle, in order to do that, needs to produce 3 times higher forces to avoid that the ankle would just twist. So that means that during standing, you have a Achilles tendon force, which is 2,100 Newton, whilst during hopping, you already have 2,500 Newtons that act on your Achilles tendon. And now if you look at the tibia again, so remember tibia equals F muscle plus F gravity. Then you have, during standing, you have the force on the tibia, which is 2,800 Newton, and during hopping, you have 10,000 Newton force acting on your tibia, which is a lot of force, which leads to a lot of strain and a lot of mechanical stress. And this is then, of course, an explanation why hopping is so efficient. We've done also some studies in microgravity and hypogravity. So we're planning something on the ISS. We have to convince our engineers, of course, that hopping on the ISS and microgravity is possible. But here, one take-home message is that the middle graph is the important one here. So in this graph, in this study, we have tested different gravity levels, ranging from 0.16 G, which is lunar gravity, to 0.72 G, which is in between lunar and Martian gravity, to 0.38 G, which is Martian gravity, to 0.7 G, which is kind of the average load that astronauts apply to themselves when they're running on the treadmill. And the interesting thing is, and this is really sub-maximal hopping, no maximal hopping. When you look at the height of the hopping, and we did this in a verticalized treadmill, so the picture on the right-hand side, so people were suspended on that treadmill and they hopped, and we measured the ground reaction forces on the left side with force sensors. And the interesting thing is, even if you look at the lunar gravity, and if you would jump in lunar gravity only 20 centimeters high, the ground reaction forces are approximately similar to standing in 1G, which is very remarkable. And if you would go to, let's say, the Mars, and you only jump 20 centimeters high, you have ground reaction forces that are twice as high as standing in terrestrial or Earth gravity. And if we go back to this graph again, this means, of course, that this will have a significant impact on the force, because if you have ground reaction forces that are higher, then this needs to be compensated again by the muscle, and ultimately this will result in very high forces acting on the tibia. So again, we can show that hopping in microgravity and in hypogravity could be, or hopefully will be, a very effective countermeasure in order to maintain skeletal integrity. Of course, when we talk about how we are going to implement this in space, there are some things that we still need to find out. The first one is, as I mentioned before, is the transmission of vibrations. Of course, every station in microgravity is very sensible and very sensitive to vibrations. So we want to avoid that vibrations, especially vibrations in a certain resonance frequency, start to shake the entire station. So we need to find ways to avoid that, and that's why we need to talk to our engineers. And of course, the left-hand side is a smaller habitat on the Loon. An idea for that. We have not a lot of space, so we need to find a way to implement this kind of exercise in a small station, or in a small lunar habitat, or in a small space vehicle like the Gateway. And of course, we don't know so much about the injury risk, because high forces, of course, they could imply that you have a higher injury risk. And of course, an injury in space is definitely something that we want to avoid. We have some upcoming studies. I'm not going into the detail about this, but jumping, just to let you know, jumping and hopping is definitely something that I'm personally very interested in, because it's a very promising countermeasure for astronauts, but also for terrestrial people in bed. If you would find the right tools to let them jump in bed, for example, something like the bottom right picture, like a jump sled that could be easily implemented in hospitals to let them jump on a daily basis in order to keep them fit and to promote bone growth, muscle growth, and also a certain cardiovascular stimulus. So with that, I'm already coming to an end. I think in order to summarize, I can say that plyometric exercise is a promising candidate to prevent and mitigate musculoskeletal deconditioning, and that's why I do a lot of research on that. Our safety aspects, they require more research, and we need to determine or we need to find the right definition of an adequate and safe dose, because we need to have a high stimulus, but we don't want to be too high to cause damage or to cause injuries. And then, the bottom is we Europeans, we've been once explorers, we will always be explorers. And I'm very sure that this applies to my fellow Indian friends, that the Indians have always been explorers and will always be explorers. And again, I was very happy to talk to you today. If you want to contact me, you can find me on ResearchGate, or you can see my email address here. If you have questions about spaceflight or European human spaceflight, then feel free to contact me, again, via ResearchGate or via email. And again, thank you very much for the invitation. It was a pleasure to talk to you. And I'm now happy to answer your questions. And I hope it was interesting for you to learn a little bit about my research and about the European spaceflight program, human spaceflight program in a nutshell. Thank you very much for your attention.
Thank you, sir. Trina, you can put in some questions for sir.
Yes, ma'am. I am checking, but there is no such questions in the chat box.
Sir, I have a question, sir.
Yes, ma'am. Please go ahead.
Sir, I have a question. I just wanted to know, you said about the other physiological injuries and the space exploration. I just want to know about the brain's function during this international space station exploration. When they come back, how they come back with their brain functions.
Okay, so we have I'm not a brain expert, I have to say, but we have no significant brain damage, I would say. So there is no cognitive impairment in our astronauts. They come back as fit cognitively as before. But there are, of course, changes in the brain that happen due to a phenomenon that we call fluid shift. So we have more fluid that flows towards your upper body because there's no gravity. And there's a phenomenon that is...
Angiogenesis. Is there angiogenesis?
Angiogenesis?
Yes.
So you mean the growth
of the vessels, more development of the vessels in the brain?
In the brain? No, there's no additional angiogenesis, but there is something that we call SANS. It's called spaceflight associated neuroocular syndrome. If you go on PubMed and Google or search for SANS, you'll find a lot of literature in that regard, and there is the phenomenon is that there's a certain pressure that acts on the optic discs, and this leads to visual impairments of the astronauts. And this is something that many astronauts experience, and which is of course a problem, because we need astronauts to be in full capacity of their visual function.
Another question: the bones of the limbs, they or the lower limbs, they become shrink. They shrink. Yeah, but what is the effect on the synthesis of erythropoiesis? Rather, erythropoiesis in the long bones. If there is shrinkage, is it a change in the erythropoiesis?
No, I don't think so. The thing is that mostly the bone that is most affected is the compact bone, and not the trabecular bone. Yeah.
Okay. And if there is a fluid shift in the upper part, that we in the case of astronauts, the upper part gets the fluid shifts to the upper part, then is there change in the heart size or size of those organs? Is there any change?
Yes, there are changes of the cardiovascular system for sure. But this is all reversible. So I would say that the body is very capable of adapting to a new environment. So initially, when the astronauts go to space, because there's more venous return, more fluid in the upper body, the cardiac output is higher. Okay. And in return, the body thinks that there's too much fluid in the body. So initially they have to go to the toilet quite often, and they reduce their blood plasma. So over time, this phenomenon, they adapt to the new environment, microgravity. And when they return to Earth, as I said before, we have something called autostatic intolerance, which is partly due to the loss of plasma volume, because they simply don't have enough blood volume to maintain brain perfusion. And that's why they tend to faint. But this is all reversible. There are no long-term consequences of that.
Another question: is there effect on how is the effect on the microbes or the gut microbiome on this due to this microgravity?
That's a very good question. I have recently reviewed a paper that was one of the very few studies that actually looked at the microbiome. I think there are some changes.
Actually, I'm trying to looking at it on the in silico model or in vitro model with a microgravity that is clinostat.
Right. Okay. Yeah, that's a very interesting topic in space. I have to say that it is kind of understudied at the moment. There are some research groups that have published initial findings, but it's a topic that definitely requires more attention to see how the microbiota changes in microgravity and what you can do about it. Because, of course, the microbiome is very important for overall health, the gut-brain axis, so for mental well-being, and yeah.
Wonderful lecture, and it's a privilege to have you in our webinar. Thank you very much. Anna?
Thank you, sir. It was our great pleasure to have you with us, and you have enlightened us about the space etiology and the countermeasure concepts which we can take. Thank you so much for enlightening us on this subject.
You're most welcome, and it was a pleasure. Thank you for the invitation, and yeah, I wish you all a successful end of the workshop. I'm sure it's going to be very interesting for all attendants, and I wish you all the best for your careers, for your personal lives, and I hope you stay all healthy and in good shape in India. And it was my pleasure to talk to you and to create a link from Germany to India, and
Yes.
also for me quite unique to have that experience to talk to a group of people in India. So thank you very much for the invitation. It was my pleasure as well to talk to
Okay,
you all.
thank you,
sir. Thank you once again.
once again.
Okay. Thank you for the attention, and again, have a nice rest of the conference, and I hope you enjoy it. And
It's the end lecture that you have given, and it's the ending, and its ending is so good, so enthusiastic, and we have a lot of viewers in our YouTube channel, more than 1,000 viewers in each day, or nearly 2,000 views in each day. So your lecture will be taken by 1,000 or more people in India and other countries also. We have different countries linked to us. Thank you very much.
that's great. Thank you very much. I have just posted my
Yes,
ResearchGate profile to the chat, so if you want to contact me, I think that will be the quickest way, and then you can also see about the research that we do in ESA, and you always see the latest publications coming from our lab. So if you want to learn more about human space research, then swing by, have a look, and feel free to contact me with a quick message if you have questions, and no problem.
Thank you.
Thank you, sir. Thanks me.
All right. Thank you all very much. Regards from Cologne, and
Bye.
take care and stay healthy. That's the most important thing.
You also take care,
You
sir.
also stay healthy in this COVID situation.
Thank you very much. Goodbye,
Thank you,
everybody.
sir. Bye, sir.
Bye.
So with this, we come to an end of our lecture session. I would also like to mention that the link of feedback form will be given in Google Meet chat box and YouTube platform. Once the validity session is over, the feedback will be received till 6 p.m. today. Now, I would hand over the mic to Dr. Annalakshmi Chatterjee, Madam, Organizing Secretary of this webinar series committee, to start the validity session. Thank you, Madam.
Thank you, Trina. Good evening once again, everyone. Thank you for everyone's attendance and attention towards our international webinar series for the second time this year. The cruise of this webinar was to engross our knowledge in the emerging areas of research in the field of food science and nutrition. That has been satisfied, I hope, with the series of lectures we had from various personalities and researchers and scientists from different areas of food chemistry, economics, food science, nutritional sciences, medicine. In all the fields we had experts, and we have been listening to their wonderful deliberations. And we come to the end of the second series of our webinar. With this, I thank you all. Pubaliti, do we have a validatory guest joined us?
Not yet joined, but he will join at 5 p.m. So I can give my thank.
Okay, you can. Now I welcome our head of the department and dean to say a few words on the valedictory.
Good evening, honorable guests, teachers, colleagues, delegates, friends, and my students, dear students. It's my privilege and honor to have been asked to propose a vote of thanks on this occasion. So I thank the Department of Home Science, University of Calcutta, for giving me the opportunity to express my gratitude on this special day. On behalf of the Department of Home Science, University of Calcutta, I extend a warm welcome to the people in the gathering. This year also successfully we have hosted one of the biggest and successful webinar series in India on the occasion of National Nutrition Month 2021, spanning about 7 days and in the presence of several world-renowned science epitomes. Department of Home Science, University of Calcutta, being one of the premier institutes, celebrates the whole month of September as Nutrition Month every year. Due to the COVID-19, we shifted to webinar series this year. I would also like to express my heartfelt gratitude to all the esteemed delegates of the 1 minute. Sorry, I had a phone call. Yeah, this event would have been impossible without the support of the faculty members, organizing committee, research scholars, and dear students of the master's fourth semester, the outgoing fourth semester. I extend my gratitude to our honorable Chief Guest, Dr. Shankar Ghosh, ex-Vice Chancellor, Kalyani University, to take time from his busy schedule to grace the event, and he will be joining very soon. Thank you for inspiring and encouraging us with your words on the special day, and he will be given a short lecture on
Ma'am, please unmute yourself.
Oh, sorry, very sorry. So, a special thanks to Dr. Purba Chattopadhyay, Dr. Anna Lakshmi Chatterjee, and other faculty members, without whose cooperation this webinar would not have been possible. I must thank the organizing team, the volunteers for working hard for the past few days to make this webinar successful. I congratulate all the poster presenters for their active participation. Thank you everyone once again for making it a great success. I want to thank the authorities, Professor Shunali Chakraborty Banerjee, our Vice Chancellor, Madam, our Pro Vice Chancellor, Sir, Professor Ashish Chattopadhyay, and Professor Debashish Das, our Registrar, for giving the chance to organize this international webinar. More than 1,000 registrations from various colleges, universities throughout India, Bangladesh, as well as other countries throughout the world, and their active participation in the webinar series is a huge success in itself. I would like to thank all the respected representatives and academicians. I, on the behalf of Department of Home Science, University of Calcutta, and the entire organization and organizing members, my colleagues, students, let me call it fraternity of food and nutrition here together, and on my own behalf extend a very hearty vote of thanks to all the speakers for gracing your important work and sharing with us your findings and opinions throughout the week. The lectures were full of knowledge and interesting things, giving deep insight into various topics such as nutritional economics, sensory biology of food, space physiology, space nutrition, nutrigenomics, computer science, and also revealed various interesting facets of those research domains. I thank you all in this occasion, and a big thank you to Professor Hemlata, Director NIN, for his gracious presence on the introductory session of the webinar series. So thank you very much. Now I hand over the mic to Anna Lakshmi.
Hey, do we need to wait for the guest, or
Yes, Professor.
he'll be joining us, isn't it, at 5? One more minute.
1 more minute. I think he will.
Okay, we shall wait for sir to join. Purvadi, meanwhile, you can say some words about the seminar. Purvadi, are you there?
Yes, I am there. I was actually waiting for sir for the word, and that I would have concluded today's deliberations.
Okay, let sir join. It's almost time.
So it has been a very beautiful journey for these past days from 1st of September to the 7th of September. Today we are on the last day, and I suppose sir has joined.
Sir has joined. Sir
Very
has joined.
okay.
Welcome, sir.
Okay. Welcome, sir. Sir, please unmute yourself.
It's okay right now?
Yes, sir.
Yes,
It's our
sir.
privilege to have you among us. It's a momentous event where, at the culmination of the things, we are having a person of your statures to grace the occasion to deliver the valedictory address, sir. This is a small department of University of Calcutta, Home Science, and we are having two disciplines. One is human development, another is nutrition, and we have been celebrating the National Nutrition Month for the week-long international webinar series where distinguished speakers from all over the world have enriched us with their diverse kind of lectures, and today we are eagerly waiting for the final panel lecture of this week-long deliberations with your valedictory address. We heartily welcome you, sir.
Thank you. Is audible?
Yes, sir, you are audible.
Okay. So, really, I'm happy to join today the international webinar on the nutrition, because India has started the nutrition week as well as the nutrition month in the September, which might be a good start. And today is a very important day also. I can just share with you why in respect of nutrition. And the last 7 days, I know that you are so busy for arranging all the good people. I have seen the the flyer. It's a really good people throughout the globe, and I really thank to Wali ma'am to organize this kind of international webinar with your small department. But it's small, but the people are very enthusiastic and might be they are very helpful to each other. That's why this such kind of webinar can be arranged. And today is the last day. Like I'm really sorry that I cannot attend any one because with my busy schedule, just 5 minutes back I came to my home. I'm anyway, and this is a offbeat to my subject that I can tell you. But and today is the nutrition is not the food and diet. It is a global chapter. It is basically the system biology and data science. Right now, the nutrition is not a core group of food and nutrition student, food and nutrition teachers. Why I am telling this thing? Because in biological science, the big project is a human genome project. And it took 10 years to complete one genome sequences. And 2003, after 50 years of discovery of DNA strand, all of we know that DNA have stated in 1953 and got the Nobel Prize, the Watson group. And after 50 years, the whole genome sequences has been published simultaneously from the UK and USA. And it took 10 years to complete a genome sequences. And today, it's very important thing that 10 years now it equivalent to 5 days. So within 20 years, how much we are processing, how much we are technologically developed, that every corner of the country of any scientific area, even in the English people, has started the genomics and got the Nobel Prize, Thomas, in 1989. He did a PhD in English. Then he thought the life has 4 letters, ATGC, how this phenotypic expression of cancer may come, and he studied very vigorously and got the Nobel Prize in 1989. Similarly, I just want to say the nutrition, that means we want to say in different category like diet, food, adulteration, like these are the common features, keywords of the nutrition. No? But to me, this is very old concept. To me, it is like a personalized medicine. It's a personalized nutrition. So person to person, the nutrition, diet, food, and behavior are completely different. That's why a malnutrition children having a same environment, somebody has a malnutrition and somebody has no. Why? Due to the personalized diet or personalized nutrition. Anyway, so I just want to share today the biggest disaster in our planet. All are very well known, and we are facing the problem. That's why we are in online. The COVID-19. On the other hand, I can tell you the COVID-19, a deadly viral disease, and infect anybody and get rid of different kind of ailment. Somebody died, somebody recovered, which is also called post-COVID. And the situation is very horrible throughout the globe. All places, all the countries are suffering this COVID. On the other hand, in every corner there is a Newton law, third law. And I can tell you one positive thing came in the nutrition field. People right now used to know the RT-PCR. Those are illiterate. They know the RT-PCR, what the things is going on. What RT-PCR, because you need to test the COVID-19 positive or negative, so make the RT-PCR. No, so this is a one kind of every corner today the molecular biology or RT-PCR. Similarly, every corner there is so many depression, mental health, and the nutrition of the COVID-19. You know, the diet of the COVID-19. So we know there are three things that we have to follow. Wear our masks, we know, wash our hand, and keep the distance. And fourth should be included, you take good food, hygienic food, balanced food, so that you can get overcome very quickly. And you can make your immunity very fast. But it was found very recently the vegetative diet or plant-based diet and the fish is very, very important for the COVID-19 area. Recently, that 3 months back, a paper has come that plant-based diet is very important for the recovery of the COVID and that. And they have given some kind of good analysis or good hypothesis about that. And similarly, if you get the mental health, the depression, presently the people, particularly the student, they are unable to go school, they are unable to go college. So every time they are facing same kind of depression, only parents and the routine job inside the 2 rooms, very fascinating or unpleasant weather in the home, in the family, that the student cannot move anywhere. So that's why we are in online. So that mental health also, that means the depression, the system, diet can also help. And there are lots of different kind of diet and the system that change the mood and that change the mood and the depression also. So these are the very important areas nowadays. The people are thinking about how diet can change the mood, how diet can change the mental depression. And people are working over there. And this is a very good area. The depression due to the disease or anything can be correlated with the diet. And on the other hand, that routine part, like a different kind of pregnancy, we have to give the special kind of food, like a folic acid, iron tablet, etc. But even though we are recommending that folic acid, etc., but how it works and how it comes to the in function to give a very good area of function or the nutritional aspect over there. So that might be a very helpful, even the different kinds of food that we are taking, and in the microbes, there are a very good role of microbes. The microbiome or microbiota present into the gut. In 2012, there is a big paper on the Nature that we have the 10 to the power 14 different microbes in our gut, which is the more than our cell. Our cell in 10 to the power 13, and more than 10% the gut microbiome in our gut. So what are their role in respect of the nutrition, in respect of absorption, in respect of conversion of the food materials? It's a very important issue, because after Human Genome Project, NIH has started the microbiome project. Every corner of this microbiome, they feel if someone is making a mental health, or someone going to obesity, or someone going to any depression, microbiome may be a role. That's why the microbes and their genetic diversity and their probiotics, etc., is very important to address the health and disease and the inflammation, etc. So microbiome is a very important issues. So that's why the globe has started the after Human Genome Project, big microbiome project. To me, after microbiome project, or parallelly, the Nutri Genome project should started in different corners in a cohort study, like a cardiovascular, cancer, diabetes, etc. etc. The cohort study have started, but in the cancer, there are lots of people are dying in the cancer. That's why the cancer started a very well versatile way throughout the globe, particularly in genomics and immunology aspect. Even in India, the cancer genomics has given a very good driven over there over the past few years. Now, come to your subject. Like I'm not a expert on that. However, my feelings I want to share with you. We know the food that contains the nutrients. We know the carbohydrate. We know the protein. We know the fat and different kind of minerals. Some minerals or some vitamins are also associated with very disease, like vitamin D, vitamin A, vitamin 12. We know, and class 7, 8 students are studying on that part. And apart from that, the micronutrients or different kind of nutrients, there are lots of other interactive molecules are there. And the nanoparticles, one of the example is nanoparticles or environmental buffer solutions over there in the cells or in the activity area, that need to be studied on that area, because the action potential of some kind of nutrients with the one cell to another cell, that makes the system biology. And this system biology is the pioneer part or in thoughts that we can go for this nutrition that makes the physics, chemistry, biology, and the mathematics, statistics, computer science, and that makes the system biology will address the entire process of the nutrients and their beyond. So what the new things are coming, the system biology interaction to the one cell to another cell, or one active molecules to another active molecules. So get in touch to each other, and how it address to any function that makes the system biology over there. I can give a very good, like a vitamins. There are lots of vitamins. Vitamin C, 6 rise in inflammation and so on. That how it makes. We are really giving that part, but how it connect to each other. What are the important areas over there that needs to part. So that's why nanoparticles and these things are very important. And today in Nature, on 7th September, a paper has come. Just 3 hours back, the paper has come. What the paper? The nutritional diversity, nutritional stability with the diversity of the crops. What are the crops we are using throughout the globe? 184 countries, the crop ingredients and their nutritional status have been published today. and in the crop diversity. So the 100 years ago or 50 years ago, there is a rice variety, indigenous rice variety. Now you cannot get this kind of rice variety. It makes the hybrid or the high yielding varieties of the rice or paddy, whatever things are there. And 55 years study and 184 is and 22,000 of different crops have been studies and published today that the nutritional stability or sustainability remains year after year. It's a good thing for us that you cannot tell that don't take this rice, that rice is containing these, because the nutritional stability have been published today. It's a very good information in the crop science. But in the animal, that we want to take some kind of animal, the diversity of the animal, we have no data in India. That fish or wheat fish or mrigel fish, what are the ingredients present over there? But in case of plant or crops, the developed country, and they have made this thing, and using the data science or machine learning process, they have made the within the supercomputer analysis. They publish this paper. But in case of animal, we do not have this kind of data that we can reciprocate. So this is the young generation. Particularly, we are lucky that we have a very huge amount of young generation. Those who are in the analysis of those kind of highly varieties of flora and fauna in Indian biodiversity, and that can be used as a food with their ingredients, and how the bad thing and good things are there, that can be analyzed, and new generation, the young generation, have to do the major part of that part. So in this content that we want to say, the coming research or we want to get some kind of good area, because we are lucky. India has a lucky because we have a lots of biodiversity. We have a lots of different kind of crops and animal food. You cannot get this kind of crops, animal foods in other countries. So how this food or how this different kind of environmental biota or microbiome that enrich our body to make to protect our inflammation and genetic cause. So we need to get some kind of research. Already have started in India, but in abroad they have already make some kind of good initiation with a cohort study, with a cohort like a cardiovascular system, diabetes, and there are lots of different cohort study of the cancer in different. I am working on the cancer since a decade, and cancer genomics. I can feel how the cancer genomics and their interaction have been developed throughout the globe. It's a very promising that we are to able to say that whether it will be a risk factor of the such kind of cancer or not, we are able to say. That's why, and in the cancer, that one mutation and there are subsequent mutation has come up in, which is called the somatic mutation, and there are well studied on this area, particularly lung cancer, oral cancer. So the mutation of these subsequent genes that are leading to cancer, and the cancer patient die with the immune deficiency. Now in developed country like US, the cancer is like a chronic disease. They won't bother about the cancer because the early diagnosis and treatment is very easier process they have developed. We are very, very behind of that because we do not have that kind of mindset or approach to execute our findings to develop these cancer areas. Similarly, in the nutrition, the nutrigenomics or genetics, which is a different term, and epigenetics, So epigenetics, that means the environmental factor that makes a change in your body or makes your methylation or acetylation epigenetically, that make a big differences. And the genetic, like I can give one example. Like we know the cow, and cow casein protein. All of you know the cow and casein protein. Have you any idea? There is a A2 milk and A1 milk and A2 milk. Hopefully, you have some idea. A1 milk or A2 milk. You cannot see the milk. Milk is white. It is coming in your market, and you can take the milk 1 liter, the 60 rupees, and you can boil and take this milk. You have no idea the milk is A1 or A2, right? And what is A1 and A2? The A1 and A2 is one kind of mutation in the cow in 67 position, the histidine that changed to proline. Histidine that changed to proline, this milk is come into your gut, they produce a bioreactive compounds, and that is very detrimental to you, and that makes the diabetes, that makes the acne, that makes the osteoarthritis, and even the heart diseases. So we are not able to know what kind of milk we are consuming. Just like you see the sickle cell anemia, if it's some kind of mutation, and this girl or boy is going to a sickle type of RBC, so they cannot carry the oxygen or carbon dioxide. And same type A1 and A2 milk, which is present in the cow, we are unable to see. Even in India, the FASA is not recognize them because there is no data. But if you go in US, A1 milk. So all their cows are characterized because the human health. So these kind of human health, there are lots of protein and lots of enzymes are producing these animals and plants. Whether this compound is detrimental to our health or not, it need to be addressed. And that's why there are lots of information is necessary to make our health is become better health. So the artificial intelligence or machine learning, the data science can help us taking all the data from the Indian content and other content and get a systematic analysis and get some kind of very good isolation or diagnosis on that part. So that's a genome. That's a one kind of environmental factors that is a circadian protein. Even though in our case, some obesity is there, there are some kind of signature or some kind of change of the gene, which might be a epio gene. One gene that is some kind of mutation may increase the obesity. If you reduce the fat, another part, it might be helpful. No carb, no obesity, but it is not always true. So these kind of personalized genetics, they say some kind of mutation in your gene that makes the change of your nutritional update or metabolism or these things. So that kind of change in the mutation in a particular gene for converting into the end molecules is restricted. That's why the genetics is a one kind of, right now there is a heart disease, there is a diabetes or any disease is related, the gene panel has been isolated. But but these gene panel are very much personalized, because Bengali people, the genetic signature is never coincide with the American, Australian, even the Europe, even the North Indian part. This is the only area the signature of your genome is a completely, or it's quite differ to the other ethnic group. That's why we need to do our own genetic information so that you can able to get some kind of good relationship over there. So now it come into the genetic, and you come into the genomics also. The genomics is a very simple, that the diet or nutrient that change your expression of the gene. Like I can say, I can give it, I have a very on that area, like a tobacco, tobacco that makes the oral cancer, so tobacco is an environmental factor. That is environmental factor and concentration of your squamous cells, and makes the change mutation and develop. But there is one gene which is called guardian of the genes, that protect the cancer. That protect the cancer. If you have reached mutation in p53, then it is better for that person to get the cancer. So if you were to get the tobacco everybody, you cannot get the cancer everybody. But it's that one to say it is a one kind of personalized. That it is a one kind of personalized. It's audible right now.
Now it
Hello.
is audible. A little disturbance. Now it's audible. Okay.
Thank you. So the genetics and genomics, the nutrient that you want to take, some kind of nutrient. That's just I'm giving some kind of one example, citing over there. In some cases, some, like all of you know, the tulsi pata is a very good health and this and that, but not in all tulsi pata or all variety. So some plant, in Nagaland, it's my personal experience that you want to see the some plant in Nagaland, that extract of this plant are using for the oral cancer, and it's a very good giving the answer. A group of Japanese, they came and took the plant and implant into the Bhubaneswar. Then they have extract this plant and it will not work to the any oral cancer. So same plant, but the GPS based plant is the answer. That's why all tulsi pata will not act your part. So this kind of micronutrient or environmental factor that interact with the cell, that makes your very good answer. So this kind of expression based on this nutrient is a nutrigenomics. So how it can increase or decrease, it is not well studied. But in cancer, there is a well studied over there, because more than 5 lakhs exome sequences have been carried out, and there are lots of people are working in the cancer genomics and their expression. So based on this observation, people are now jumping the core study of the nutrition, whether the cardiac disease or in the renal disease or the diabetes. So there are lots of research based on this genetic and genomics have already started on the cohort basis. And after that, the environmental factor, the HPV that causes the cervical cancer. So HPV is an environmental factor. Tobacco is an environmental factor. And in the Kashmiri, there is a belly cancer. Most of the women are suffering from the belly cancer because they want to put the hot on the belly, and it touches the belly and make the constant irritation that makes a belly cancer. So these are the environmental factor that makes the different kind of cancer, So this is the constant irritation that change your genetic part. So it is not heritable. You cannot herit the gene from the parents, but the environment can induce you to make some kind of somatic change, and the expression of the gene might be changed because there is some methylation or acetylation that makes the change of this expression of the genes, and that becomes a different kind of diseases. And similarly, there are not much any report I can say how the methylation or acetylation or environmental factor that gives some kind of diabetes or some kind of obesity or some kind of renal failure. We have not good data on that part. But I'm 100% confident. Last 35 years, I am working on ATGC, that is DNA molecules. I'm confident that all the course here are governed by these 4 letters and their orientation. So either inherited or changing in the environment factors, which is called epigenetic or environmental genetics. So that makes interact with each other and help with the system biology and predict the personalized nutrition. And this personalized nutrition that can cope up with the genomics and the proteomics, metabolomics, and integrated with the environmental factor microbiome that makes the data analysis that help the personal nutrition. And that might be the good future. Everybody, I'm sure now people are jumping to get some kind of nutritional status or nutritional genomics or genetics that make interaction to the diabetes, that make interaction to novel disease or any chronic disease, because all of the diseases or all of the inflammation we can correlate with this ATGC or 4 letters, or particularly in genomics, proteomics, or metabolomics. So that makes a integrated analysis, and which are the personal nutrition, because we know the personalized medicine. And in US, the people are working on giving the treatment over there. If you get the paracetamol for remove your fever, and it will work for you and it may not work for you. And after 20 years or 10 years, you can get a kidney failure because it is a personalized medicine. We do not know in which pathways it works and which blocks over there. So it's a kind of a system biology that help us to understand on there. So next 20 or 30 years, we want to develop this kind of nutrition-based, some kind of omic studies, so that we can get the better personalized nutrition, all of you. And we are the luckiest country that I can tell you. We have lots of young youth, those who are interested to work in this field. Secondly, we have lots of bioresources, lots of food, and their combination that might be helpful to get our country become a unified and বিশ্বসেবায় জগৎ শ্রেষ্ঠ একদিন ভারতবর্ষ হবে। With these few words, আমি শেষ করছি এখানে। ধন্যবাদ ম্যাম। Thank you.
Thank you so much, sir, for your such a broad kind of a lecture, which actually summarized all the kinds of findings under the field of nutrition. What a way to end our international webinar series. We honestly thank you and would love to hear you in future also, and in person. Due to COVID, we could organize this kind of a virtual thing, which is a positive thing also to gain knowledge. but it would be a pleasure to have you at our department in person. Thank you so much, sir. Ubalidi?
Thank you.
I suppose Kubalidi is having some kind of a problem. So, sir, thank you so much. And with this,
Thank
our
you very much.
okay, okay.
And I requested just 1 or 2 days back, and sir has accepted my invitation. Thank you very much, sir.
Kubalidi, we
And
are very lucky. We are very lucky that we are
lucky.
getting the blessings of such great persons. Well, these are actually very lucky.
Thank you, sir.
So, with this deliberations, our webinar series comes to an end. And this is our moral duty to extend a vote of thanks. First, my vote of thanks will go to our valedictory speaker. Thank you so much, sir, for sparing your valuable time for us. And next, my thanks will be to our Vice Chancellor, Madam, our Pro Vice Chancellor, sir, our Registrar, sir, for their necessary permission for giving us the uh, permission to conduct such webinar series. Also, it is a great privilege to have so many speakers from all the corners of the world who have agreed to grace this department, although virtually. And last but not the least, this wonderful team of organizers. We are very, very lucky to have such dedicated scholars and students, one and the only Niloy, who had been instrumental in bringing all those beautiful, brilliant minds, just contacting them and again in relentlessly just try and bringing them here. And then Shomam, Trina, Shrija, Orpita, Beepsha. I hope I'm not leaving. Oh,
Rita,
Amrita. Ah, yes, Amrita. This brilliant people who have been so effervescent. Their energy was had such a spillover effect on us. Actually, it was their energy which energized us. You are our foundation, and we look forward to see you scale further heights. And of course, our participants who are the main what to say energy or the main source for which all this effort had been taken. We would like that this webinar series with your motivation, and if you want it, then maybe we can continue it in future also, hopefully not virtually. We would like to continue with the good work. And of course, at last, a special mention to our loving HOD and Dean, whose these are all her brain waves. Actually, she loves all these academic endeavors, and it is her what to say, her nature to continuously encourage her fellow colleagues, be it her students, her scholars, to continuously motivate and do good work. Madam, you are an inspiration, and we hope to see such great endeavors in our department. And with this word, I would like to put a curtain call on the entire happenings for the entire week-long events. Thank you all so much.
thank you.
Just a small announcement before we all quit, that is regarding the certificates. It has been given in the Telegram group also. Because of the large number of registered participants, it will at least take 2 weeks, if not more, to distribute the certificates. But be rest assured, those who have participated and filled up the feedback form, they will be getting the certificates, but we need a bit of time to organize things at our end. And of course, the poster presenters, those who have presented the posters, they also will get the certificates. Don't worry about it, but please bear with us and give us some time. Thank you all so much.